Namaskar. A very warm welcome in today's session on toxicity assessment. In today's session, I, Professor Shachisha from School of Interdisciplinary and Transdisciplinary Studies, IGNOU, will discuss overview of toxicity assessment, routes of exposure, toxic effect like acute effect, chronic effect, dose response assessment, dose response curve, LD50 and LC50, and finally assessment of toxicity. After completing this session, you will be able to understand toxicity assessment and the routes of exposure. You will also be able to understand the dose response relationship, lethal dose, lethal concentration, and various methods for toxicity assessment. To begin with, toxicity may be expressed in a variety of forms depending on the chemical which is involved, the nature of the population exposed, and the conditions of exposure. We can define toxicity as the ability of a substance to cause adverse effect in living organism. This ability to cause adverse effect is dependent upon several conditions. For example, it will depend on the quantity or the dose of the substance and whether the effect of the chemical are toxic, non-toxic or beneficial. In addition to dose, other factors may also influence the toxicity of the compound, such as the route of entry, duration and frequency of exposure, variation between different species, and variation among number of the same species, which we can say interspecies and intraspecies. The potential hazard to human may be acute, subchronic and chronic. The more specific type of toxicity that are determined include carcinogenicity, teratogenicity and reproductive toxicity, mutagenicity and neurotoxicity. Toxicity testing is necessary to provide some basic for regulation of substance those humans and other living things come into contact. It is used to determine the safety of range of chemicals used, for example, cosmetics, pharmaceuticals, food additives, pesticides, chemicals, additives and consumer products. A toxic effect can result from a natural or a manufactured substance and manifest a variety of symptoms which may be both immediate and long term. Toxicity testing introduces a variety of methods and rates of exposure to the test organism, which help in formulating a more accurate assessment of the risk of harm that the test substance may pose to human health and the environment. Toxicity assessment, in other words, is characterization of the toxicological properties and effect of chemical with special emphasis on establishment of dose response characteristic. Having said this session will focus on toxicity assessment and role of toxicity assessment in toxicological study. First, I will give you the overview of toxicity assessment. As I have told, the toxicity term, it refers to the inherent potential of a substance to cause systematic damage to living organism. Basically, if you say all substances are toxic in quantity. Many therapeutic medications are acutely toxic, but beneficial only when used at an appropriate level. For example, vitamin D, table salt, oxygen and water are toxic if it is taken in high quantity. Thus, the mere presence of a substance does not automatically imply harm. This is why toxicity assessment is concerned with the type and degree of harm caused by different amount of substance. In other words, I can say that a toxicity assessment is a tool to investigate the potential for a substance to cause harm and how much causes what kind of harm. Toxicity assessment is a major component of risk assessment. It provides information on how much of a chemical causes what kind of harm. Basically, toxicity assessment is based on animal study. The degree of harm to human must be extrapolated using mathematical models based on a variety of assumptions. Thus, the toxicity assessment provides only an estimate of the harms to human. As more toxicity studies on a particular chemicals are conducted, dose response studies on different species of animals. For example, epidemiological and in vitro studies, scientists become more confident in their characterization of the toxicity of the substance. Now, I will discuss with you the routes of exposure of various chemical or toxic compound. There are many routes by which a substance can enter into the body. The major routes I will be discussing in next few slides or few sessions. First is inhalation. The major routes of entry for most of the chemical in the form of vapors, gases, mist or particulate is inhalation. Once we inhaled, these chemicals are either exhaled or deposited in the respiratory tract. The deposited chemicals can damage through direct contact with tissue, or the chemical may diffuse into the blood through the lung-blood interface. The second way is absorption. The absorption of the chemical may be entered either through the skin, which is called dermal, or through eye. Dermal contact can cause redness or mild dermatitis or more severe effect like destruction of skin tissue or other debilitating condition. Many chemicals can also cross the skin barrier and be absorbed in the blood system. Once absorbed, they may produce systemic damage to internal organ. The third way is transformation. Many chemicals are metabolized or transformed via chemical reaction in the body. In some cases, chemicals are distributed and stored in specific organ. The next route of entry is ingestions. Chemicals that are inadvertently get into the mouth and are swallowed do not generally harm the gastrointestinal tract unless they are irritating or corrosive. Chemicals that are insoluble in the fluids of the gastrointestinal tract, for example, stomach, small and large intestine, are generally excreted. Others that are soluble are absorbed through the lining of the gastrointestinal tract. They are then transported by the blood to internal organ where they can cause damage. The next route of entry is injection. Substance may also enter the body if the skin is penetrated or punctured by contaminated object. Effect can then occur as the substance is circulated in the blood and deposited in the target organ. The next is storage. Storage may reduce metabolism and therefore increase the persistence of the chemical in the body. The various excretory mechanism, for example, we know these are exhaled breath, perspiration, urine, feces or detoxification. These are the method to get rid the body toxin over a period of time of various chemicals. For some chemical elimination may be a matter of days or months. For other, the elimination rate is so low that they may persist in the body for a lifetime and cause deleterious effect. Now, I will discuss the toxic effect. The toxic effect is defined as an adverse change in the structure or function of an experimental animal as a result of exposure to the chemical substance. Such changes may be affected, they may be acute, they may be subchronic or they may be chronic exposure. Acute toxicity, if we say it, this test measures the immediate effect of exposure with an estimated time for a peak effect of approximately 8 hours after the initial exposure. Toxicity test usually it focus on first cytotoxicity, which is known as cell damage, mutagenicity, which alters the genetic material, carcinogenicity, which causes cancer, teratogenicity, which causes birth defect. The effect of all these can be classified as either acute or chronic. The acute effect, it happens very rapidly after a single exposure has occurred. For example, food poisoning, breathing fumes from a chlorine spill or other spills. Sweating, nausea, paralysis and death are examples of acute effect. In acute effect, there is a rapid and serious response to a high but short-lived dose of toxic chemicals. Acute poisons interfere with essential physiological processes, leading to a variety of symptoms of distress, and if interference is sufficiently severe, it leads to death. Acute toxicity is, if we see, it is relatively easy to gauge. At high enough level, the effect of toxin or bodily functions are obvious and fairly consistent across the individual and species. These levels vary enormously for different chemicals. Almost everything, as I have told you, is toxic at some level, and the difference between toxic and non-toxic chemical is a matter of degree. The most widely used index of acute toxicity is LD50, lethal dose 50, and the lethal dose 50 is for 50% of our population. I will discuss LD50 in detail. The LD50 is obtained by graphing the number of deaths among a group of experimental animal, usually we take it rat, at various level of exposure to the chemicals and interpolating the resulting dose response curve to the dose at which half of the animal die. That is why it is known as LD50, that is 50%. The dose is generally expressed as the weight of the chemical per kilogram of body weight on the consumption, that toxicity scale inversely with the size of animal. The second type of effect is chronic effect. Chronic effect happen only after repeated long-term exposure. For example, cigarette smoking, eating foods with low level of contaminants, breathing polluted airs, etc. Cancer, organ damage, reproductive difficulties and nervous system impairment are example of chronic effect. In a chronic effect in which the dose is relatively low but prolonged and a time lag occurs between initial exposure and the full manifestation of the effect. Chronic toxins have more subtle effect, often setting in motion a chain of biochemical events that lead to disease states, which also include cancer. Chronic effect, if you see, they are much more difficult to evaluate, especially at the low exposure level that are likely to be encountered in the environment. Chronic effect, they fall into two categories: carcinogenic effect and non-carcinogenic effect. The example of non-carcinogenic effect are organ damage, reproductive difficulty, nervous system impairment, etc. Talking about organ damage, cirrhosis of liver from long-term alcohol consumption, emphysema from long-term tobacco smoking leads to organ damage. Likewise, decreased fertility from the pesticide, for example DBCB, is reproductive difficulty. Mental retardation in people exposed to high level of lead during early childhood, it leads to nervous system impairment. Now, I will discuss the dose response assessment. The amount of a chemical that an organism, maybe a person, is exposed to is called dose, and the severity of the effect of that exposure is called the response. Dose and response. A dose response assessment is a scientific study to determine the relationship between dose and response and how much dose is correlated with how much response. Response in this way can be expressed as measured or observed incidence, percent response in group of subject or population, or as the probability of occurrence within a population. The process of characterizing the relationship between the dose of an agent administered or received and the incidence of an adverse health impact in exposed population, and estimating the incidence of the effect as a function of human exposure to the agent. It also takes account of intensity of the exposure, age pattern of the exposure, and possibility of other variables that might affect response for it, such as sex, lifestyle, and other modifying factor. A dose assessment response usually require extrapolation from high to low dose and extrapolation from animal to human. The second step in the assessment of risk to human from potentially toxic agent, in which the dose relationship or relationship between the dose level to which animal or human are exposed and the health effect response at each dose level are characterized quantitatively. Often the close response assessment is based on the high dose experimental animal studies and applied to human who are exposed at much lower doses. The process of characterizing the relationship between the dose of an agent administered or received and the incidence of an adverse effect in exposed population and estimating the incidence of the effect as a function of human exposure to the agent. Now, I will discuss the dose response curve. The dose response curve is a mathematical relationship between the dose administered or received and the incidence of adverse health effect in the exposed population. Toxicity values are derived from this relationship. The characteristics of exposure to a chemical and the spectrum of effect caused by the chemical come together in a correlative relationship that toxicologists call the dose response relationship. This relationship is the most fundamental and pervasive concept if you see in toxicology. If you want to understand the potential hazard of a specific chemical, toxicologists they should know both the type of effect it produces and the amount or dose required to produce that effect. The relationship of the dose to response, it usually illustrated as a graph, and this graph is known as dose response curve. There are two types of dose response curve. One that describe the graded response of an individual to varying doses of the chemical, and other which describe the distribution of responses to different dose in a population of individual. The dose is represented on the x-axis and the response is represented on y-axis. An important aspect of dose response relationship is the concept of threshold. For most type of toxic response, there is a dose called a threshold, below which there are no adverse effects from the exposure to the chemical. The human body has defense mechanism or defenses against many toxic agent cell in human organ, especially in the liver, kidney. They break down chemical into non-toxic substance that can be eliminated from the body with urine or feces. In this way, the human body can take some toxic compound, the dose which is below the threshold, and still can remain healthy. The identification of the threshold beyond which the human body cannot remain healthy, it basically depends on the type of response that is measured and can vary depending on the individual being tested. Threshold basically it is based on acute responses such as death. They are more easily determined, while threshold for chemical that cause cancer or other chronic response are usually harder to determine. Even so, it is important to identify a level of exposure to a chemical at which there is no effect and to determine threshold when possible. If you see this figure demonstrated an S-shaped idealized dose response graph. It is typical of most type of exposure. In this figure, you can see the percentage responding is plotted against the dose in milligram per kg. This S-shaped curve illustrated that at low doses there is little or no response, while at high dose all individuals respond or demonstrate the effect. The line drawn at 50% determine at what does 50% of the population will demonstrate this response. This is a theoretical curve. In practice, such Gaussian curve is rarely found. Curve of this kind form the basis for the determination of the LD50 or LC50, that is median lethal concentration. Now I will be discussing LD50 and LC50. LD50 or lethal dose 50 is a standard measure of the toxicity of a material that will kill half of the sample population of a specific test animal in a specified period through exposure. The exposure may be either through ingestion, skin contact or injection or any other way. LD50 is measured in microgram or milligram of the material per kilogram of the test animal's body weight. Lower the amount, more toxic is the material. It is basically used in comparison of the toxicities. LD50 value cannot be directly extrapolated from one species to other or to human. Also called median lethal dose LD50, originally it is abbreviated as DL50 for dose lethalis, is 50% for a test used in animal experiment. It was designed by the British pharmacologist J. W. Treven in 1927. In other words, we can say that LD50 is the dose of any substance tested required to kill half of the number of test animal. The test shows how much of a substance must be taken before it becomes deadly. A reported rat oral LD50 or 50 mg per kg means that half of the rats that ingested a dose of 50 mg of the substance per kilogram body weight died within 14 days. The LD50 test is used to determine the acute toxicity of a substance. This is the dose at which the test substance is lethal to 50% of the test animal. During the test period, the animal forcibly inhale, ingest or is otherwise exposed to the substance. LC stands for lethal concentration. LC value usually refer to the concentration of a chemical in air, but in environmental studies it can also mean the concentration of a chemical in water. According to the OECD or Organization for Economic Cooperation and Development guidelines for the testing of chemical, a traditional experiment involved group of animals exposed to a concentration or series of concentration for a set period of time. Usually it is 4 hours. The animals are clinically observed for up to 14 days. The concentration of the chemical in air that kills 50% of the test animal during the observation period is the LC50 value. Other duration of exposure, which versus the traditional 4 hours, may apply depending on specific laws. The LD50 and LC50 are specific cause of the generalized value LDN and LCN. The LDN is the dose of a toxicant lethal to n% of toxic population. The LCN is the exposure concentration of a toxicant lethal to n% of test population. Thus, the LD50 is the statistically derived single dose of a chemical that can be expected to cause death in 50% of the given population of organism under a defined set of experimental conditions. Similarly, the LC50 is the statistically derived exposure concentration. Other important value that may be derived from the relationship, it shows that the threshold dose or concentration, the minimum dose or concentration required to produce a detectable response in the test population. The threshold value can never be derived with absolute certainty, and therefore the lowest observed effect level, that is known as LOEL or the NOEL, have normally been used instead of threshold value in deriving regulatory standard. In assessing the significance of LD50 or other toxicological value, it is necessary to note that the units used in expressing doses. Normally doses is expressed in mg per kg body weight, but it may be expressed in mg per centimeter square body surface area, as this has been shown in a number of cases to permit more accurate extrapolation between animals of different size. Now I will discuss assessing toxicity. All quantitative toxicity assessment are based on the dose response curve. With the increase in the dose, which is known as exposure, the response, that is in the form of toxicity, also increases. The smaller the dose needed to cause an effect, the more potent or toxic the substance is. For all compounds other than cancer causing agent, which are known as carcinogens, it is assumed that there is a dose below which no effect occurs, which is known as a threshold. This is similar to a drug where too small of a dose has no beneficial effect. For carcinogens, it is often assumed that even the smallest dose can cause effect. Although the dose response concept is used in all type of toxicity assessment, it is used somehow differently for each of them. The toxicity assessment commonly involve two steps: hazard identification and dose response assessment. The outcome of a toxicity assessment is usually expressed as a toxic value, such as a reference dose RFD or cancer slope factor CSF, which incorporate the finding of the hazard and dose response assessment and safety factor that address uncertainties in the assessment. A toxic assessment may also include that a toxicity value cannot be developed because of inadequate data. Acute toxic effects are estimated by LD50 studies or observation of accidental exposure. Acute toxicity is assessed using observation of accidental human exposure or by conducting LD50 test on experimental, usually rodent. Chronic toxic effects are estimated by dose response studies on animal. Carcinogenic effects are estimated by a type of dose response study called a carcinogenesis bioassay. In the toxicity testing, the toxicity can be evaluated in whole organism, that is in vivo, or using molecules or cells, that is known as in vitro. The main advantage of toxicity testing is that it detects toxic compound based on their biological activity, and as such does not require a prior knowledge of the toxicant to identify its presence. The same characteristic is also have a disadvantage, because while toxicity testing can determine if toxic compounds are present, it does not identify them. Identification of the toxic compound is then required. Once a suspected toxicant is identified, modeling approaches, which is known as in silico approaches, can sometimes be used to predict its toxicity based on the physicochemical properties of the compound and its likely fate and transport in the environment. Now I will discuss the direct toxicity assessment and indirect toxicity assessment. The direct toxicity assessment can be assessed, number one, by in vivo bioassay. These are conventional toxicity testing which relies on direct toxicity assessment of whole organism, whether it is alga, shrimp, sea urchin, fish, rat, etc. In this, the organism are exposed to the chemical or mixture of chemical and monitored for any sign of adverse effect. This can be either a gross morphological effect, for example, weight loss, visible lesions, death, or more subtle biochemical markers. These being neither biomarker of exposure or biomarker of effect. For example, biomarker of exposure are indicator of the internal dose such as metabolite in urine, and biomarker of effect are an indicator of health effects such as enzyme activity. The duration of the exposure depends on the type of toxicity detected or being monitored, from short term acute effect, which is 96 hours or less, or subacute effect, which is a couple of days, or subchronic effect, which is a couple of weeks, through the chronic effect, which is a significant portion of the organism life expectancy. Depending on the species used, in vivo toxicity testing is generally seen as the most relevant predictor of human health effect. This is because in vivo tests include a measure of absorption, distribution, metabolism and excretion, all of which could modulate the toxicity of the sample. The second direct method is in vitro bioassay or bioanalytical method. In vitro bioassay have been in use for drug discovery by the pharmaceutical industry for decades. In vitro bioassay, molecules, for example enzymes or a whole cells are exposed to chemical or mixture of chemical of interest and they are monitored for specific response. There is more to health than cellular health and human beings are more than simply billions of independent cells. However, for chemically induced toxicity, the initial interaction of chemical at the molecular or cellular level is necessary prerequisite for toxicity. This is because toxicity occurs at the site of interaction of the toxicant, which can be either the parent compound or a metabolite, and the target biomolecule, which is known as primary effect. Organisms, however, have defense and detoxification mechanism to cope with certain degree of primary toxicity, and it is only when those defense mechanisms are overcome that observable toxicity occur, which is known as secondary effect. Typically, in vitro tests are carried out on a specific cell type depending on the endpoint of interest. Some assay can be more variable than others, and through quality assurance, quality control procedures such as consistent use of positive and negative control, monitoring of assay performance with the control chart, quantification of detection limit, determination of reproducibility and robustness, use of inter-assay sample, intra and inter-assay duplication and adoption of good laboratory practice help ensure the production of reliable high quality data. Each type of bioassay has its advantage and limitation, and no single assay can provide a complete assessment of the biological activity of a sample. Therefore, a battery of bioassays is required to rigorously assess the potential of a sample to cause biological effect in exposed organism. In direct toxicity assessment, the first I will discuss in silico approach. Some of the shortcomings of an in vitro bioassay, particularly the lack of integration of toxicokinetics, can be partially overcome by combining them with computer, that is known as in silico computer modeling using structural activity relationship, that is known as SAR. In SAR, the chemical structure and other physicochemical properties of the substance can be used to predict its toxicokinetics. In silico methods are very useful in the absence of other toxicological data, but are based on data from other chemicals and such should be viewed with appropriate caution. If it assess the acute toxicity test, acute toxicity standard test, though appears to be simple, routine method, but they should be performed strictly in accordance with standard requirement, observing recommended condition and using indicated organism. The tests are conducted either to determine the concentration of a test material, whether it is a chemical or effluent, or the level of an agent, whether it is temperature or pH, that produces a deleterious effect on a group of test organism during short term exposure until control condition. These tests are performed to determine the concentration of a particular chemical that would elicit a special response or measurable endpoint from a test species in a relatively short period of time, usually from 2 to 7 days. These tests are also applied in order to compare the sensitivity of different species or organism at different stages of their development to find the relationship with other variables that affect overall results. These tests must meet the following requirement. They must be applicable to a wide range of chemicals. They must be short, reproducible, sensitive, cheap and of low variability. They must be standardized, accepted by the regulatory and scientific community. The assessing chronic toxicity are more complex and the effects are studied for a longer period of time. The aim of these tests is to determine the concentration of a test material or substance that produce an adverse effect on a group of test organisms during long-term exposure under control condition. Usually, chronic tests are conducted with a set of species of different phylogenetic level and different stage of development. Unlike acute toxicity test, chronic toxicity studies evaluate not only mortality but also investigate endpoint such as individual growth rate, abnormal development, hatching time and success reproduction, vitality of offspring, behavior of individual, physiological parameter or histological. Using these methods, NOEC and LOEC can be calculated. As I have told you, NOEC is no observed effect concentration and LOEC is the lowest observed effect concentration. Chronic toxicity can be divided into two categories, that is cancer causing or carcinogenic toxicity, all other effect that is non-carcinogenic toxicity. Cancer is a separate category because public concern about it is so great. People want to know if even one person in a million person who are exposed to a substance will get cancer. To discover this, researcher conduct a specific type of dose response study which is known as carcinogenesis bioassay. The non-carcinogenic effects are usually assessed with a different type of dose response study. The non-carcinogenic assessment is assessed by studies to determine the smallest dose that cause any detectable effect. Scientists across non-carcinogenic chronic toxicity by administering various amount of a substance to laboratory animal and noting the effect if any of each dose. Carcinogenic bioassay, in this scientists assess carcinogenic toxicity very differently than they assess non-carcinogenic toxicity. This is in response to public fear about the cancer. The impracticability of such experiment has led to development of many carcinogenic bioassay. Usually scientists are not looking for the safe level of exposure. This is a method of testing substance for carcinogenous effect and it should be based on various scientific practices. If I say the example of carcinogenic bioassay, they are performed for example for benzene on both rat and mice. Both sexes of each species got leukemia at the high dose administered. Extrapolating the cancer incidence at high dose to low dose and from rodent to human resulted in the risk estimate that a benzene dose of 1 milligram per kg per day will result in 3 cancers per 100 people exposed daily for a lifetime to that dose. This dose is much higher than anyone would be exposed to in the environment under normal condition. So, to conclude this session, I can say that a toxic effect can result from a natural or manufactured substance and manifest a variety of symptoms which may be both immediate and long term. Toxicity testing introduces a variety of methods and rates of exposure to the test organism, which help in formulating a more accurate assessment of the risk of harm that the test substance may pose to human health and environment. Toxicity assessment is characterization of the toxicological property and effect of chemical with special emphasis on establishment of dose response characteristic. The guiding principle of toxicity testing is to check the effect of test substance on a laboratory animal and its direct toxic effect on human. And secondly, the exposure of laboratory animal to high dose in order to evaluate its possible hazard to human that are exposed to much lower rate. Thank you.